In this video, we will look at how the membrane keeps things in and out of the cell. We will look at the function of the membrane and its components. The function of membranes is to keep the inside in and the outside out. The cell is surrounded by the plasma membrane. The nucleus and the mitochondria are both surrounded by a double membrane. The endoplasmic reticulum is a large membrane that folds back and forth in the cytosol of the cell. The Golgi is a series of dynamic membrane stacks. The lysosomes have a membrane, and it is a good job that they do, as they are loaded with digestive enzymes which would otherwise digest the contents of the cell. The peroxisomes have a membrane. And finally, the trans-Golgi network and the endosomal system both have membranes which are dynamic in terms of sorting and regulating and processing proteins and targeting them to the correct membranes. There is a lot of membrane and membranes in the cell. So what are all these biological membranes made of? Well, they consist of a layer of lipids and protein molecules with roughly 50% of each. Membranes form an impermeable barrier to water-soluble molecules and consist of a bilayer of lipid molecules with some associated proteins. The lipid molecules in the membrane are amphipathic, which means that they are hydrophilic, water-loving, and hydrophobic, water-hating. The head of the lipid molecule is hydrophilic and is on the inner and outer faces of the membrane, whilst the hydrophobic tails form the central core of the membrane. In an aqueous environment, because of the properties of the lipid molecule, the lipids will either form a myocyl or a lipid bilayer. That is, in an aqueous environment, the hydrophobic tails will hide from the water. If we have a lipid bilayer in an aqueous environment, then it spontaneously forms a sealed balloon of membrane, which is often referred to as a vesicle. It is important that you don't think of the lipid bilayer as being static. It's not. It has fluidity. We can get lateral diffusion and rotation of the lipid molecules in the membrane and a certain degree of flexing of the lipid tails. We can also get flip-flopping between the different sides of the membrane. However, this is rare. Now, some materials, such as small hydrophobic molecules, for example oxygen, carbon dioxide, nitrogen and steroid hormones, can easily diffuse across a membrane. But small uncharged molecules such as water, urea, glycerol and ammonia, large uncharged molecules such as glucose and sucrose, and ions cannot easily cross the membrane barrier, if at all. So this is great. We have a membrane that forms a barrier around the cell, the plasma membrane. And the membrane can prevent water-soluble molecules from entering or leaving the cell. The membrane is keeping the outside out and the inside in. However, it is also keeping some material inside the cell, such as metabolic waste, that the cell will need to remove. And it's also preventing some material from entering the cell that the cell needs to live. Inside the cell, the membranes around the different organelles are keeping the material of the cytosol in the cytosol and the material of the organelles in the organelles. And this separation or segregation of areas of the cell means that the cell can set up different environments to carry out and perform different tasks. However, there is a problem. How can a cell get material that it needs into the cell and other material out? That is, how can the cell move things across the plasma membrane or the membranes around the organelles? And what are those proteins I mentioned earlier doing in the membranes?